This is the Skin Science Podcast. I'm Dr. Thomas Hitchcock, and here we'll investigate everything skin science and dissect it from a scientific perspective, analyze it from a medical perspective, critique it from a consumer perspective, and give insight from an industry perspective. Welcome back to the podcast. I'm Dr. Thomas Hitchcock, and with us in the studio today we have the ever delightful Angela McDonald. How are you, Angela?
I'm great. How are you, Thomas?
I am okay. And we have with us also Dr. Brian Jones. How are you doing, Brian?
Doing well, thank you.
All right. So it's the summer months now. It's getting warm. Although Angela decided to wear winter clothes, I am in my
it's a cotton
spring,
jacket.
summer
It's cotton.
attire.
Summer cotton jacket.
And I mentioned that because it's getting a little warm. And today we're talking about a couple of things. The first episode today we're going to be doing on exosomes. So we'll talk about that. The second one's on hyperhidrosis, and so that's all about sweating. And that's something that I am an expert in.
Oh, my goodness. Wow.
I am a sweater.
Divulging
I am
today.
a sweater. But the first topic is exosomes, and I wanted to kind of put that into context about, it's a hot topic right now. Everybody seems to be talking about all the conferences we go to, all the practices that we talk to. It seems like everybody knows the word, but I just kind of get this feeling that everybody knows the word but doesn't really understand the word necessarily at the level that I think we should in the medical practice type of, location. What am I trying to say? The medical practice field. Yeah, the medical field. And so why don't we just start out by talking about what are exosomes? Let's define them because there is, I think, some kind of analogies that have been used about FedEx packages and all that stuff. While that's great, and I know that everybody likes it when we use analogies and not make it really technical, I do think, and you know that I feel this way, I don't like it when people become reductive.
Yes.
I hate that word, but it is a word I use a lot.
It is.
Because when you know a certain thing and in the technical field and somebody brings it to a place that's understandable, people get it and they run with it. And that can lead to problems. It's one thing to just say that to a consumer maybe, but to a physician, you want to make sure you are being somewhat accurate in your analogies. And so I'm going to let Brian give us his give us the most technical, explanation.
And then it's okay. We're going in.
It could be really.
I want it to be super technical, so everybody brace yourselves.
Okay.
Yes.
All right. But then what we'll do is then Angela,
Then we'll pass it to Angela, and then it'll go really not technical.
Well, I want Angela to say what you heard. Like, what did you grab
Very
from
good.
that?
I would love that.
You also know.
I know a
most of
little
what it
bit
is.
about exosomes, but certainly no expert on this subject. I'm looking to learn today as
Great.
well, so this is going to be great.
Great. And then once you give us your kind of reflection, and this is the reason I'm doing this, because I want to kind of see that exercise of when you explain something, what are people hearing? Mm-hmm. And we'll go from there. Okay.
Okay.
So
I love it. Let's go.
Okay. Well, these are small little membranes, little packages, so to speak, like you mentioned before.
No analogies. Make it technical.
Analogies,
Make it technical.
Again, that are membrane microvessels that are between roughly around 30 nanometers to about 160 nanometers. So again, they're very small, very tiny. But within those particular membranes are packaged materials inside which actually allow communication between different tissues, different cell types, and they can go over long distances throughout the body. And again, they sell or they provide communication to other areas regarding the condition or the situation for the tissue or the body. It's not very accurate or very detailed, but again,
well, let's make it even a little more technical before we go to Angela, because you mentioned these are little vesicles. So basically, let's just start with even cell biology. So we have cell biology where the human cells are basically encapsulated in this membrane, right? This phospholipid membrane that has all different proteins in it and lipids and cholesterol and all that stuff. Mm-hmm. And so where do these exosomes come from? What are these vesicles come from, and how are they? There's different classes of these vesicles.
Right, it's sort of based on size and what they do. But again, they're all based off of the membranes that are found in the endoplasmic reticulum. As far as taking those membranes and then they piece those out. They're then built within the cell and then released through blebbing, I guess, if you want, without details. Essentially, it forms a vessel inside the vessel and then goes through that membrane and then is excreted out of the cell, and so that actually has a package.
So I think it would be different than blubbing, right? Because blubbing is when you pinch off a piece of where this is actually exocytosis, where it's actually opening up and then releasing, right?
That's correct.
So it's different than a bleb. I just want to be clear.
It opens at the membrane, but so again,
a bleb would be more like when a cell's dying and it's releasing pieces.
Yeah, this actually is a separate membrane. It doesn't take the outer cell membrane. So again, in that particular case, it's not blubbing because it doesn't take the outer cellular membrane.
That's right.
It's actually a membrane that's inside that's then excreted.
And why is that important?
Why is that important? Well, then it actually takes interior material, and it can be totally unique between cell types, because again, it is characterized within the cell, not actually within the outer membrane of the cell, and those markers and those receptors and the like. It can be totally separate from what that cell of origin is from, are based on.
So the membrane is independent from the membrane of the cell because it's constructed specifically for that particular vesicle.
Right.
And so the reason we're getting into all this is, we'll get into in a second, but what everybody just heard, if you're not a cell biologist, if you're listening to this as a layperson, the reason I'm wanting to get into this granular detail is because I want people to kind of appreciate that there's a lot to this. And that when people are as cavalier as saying an exosome is an exosome, and using it kind of like we refer to a Kleenex, all Kleenex are not a Kleenex brand, and it's a generic. Exosome is a generic term, but there's nuance to, even within the exosomes, there's differences. And so to refer to exosomes and assume they're all the same is like my whole thing of saying all Canis lupus, all wolves are the same. No, some are beagles and some are gray wolves, because that species has very big differentiation. There may not be as big a difference with some exosomes, but maybe there is.
Well, actually, it probably is quite a bit different in regarding that. And what they've seen so far in regarding looking at the characterization, and that really will get into other things, I think,
like the markers on the cell surface and such.
I think that'll get a little bit later on as far as how those might have benefit or may have detriment in regarding the tissue response. Because again, all cell types, at least as much as we know, produce exosomes. And again, these are normal cells. These are cancerous cells. Again, cells under stress, cells that are growing. I mean, so there's all sorts of different conditions which may produce a exosome, and those may have different characterizations or different packages of material that send a signal to other cells.
Right. Now, the one thing that I think people also need to appreciate is that a lot of the information about exosomes that is published is done in vitro situations, which is different than in situ, which is a fancy way of saying in a petri dish or cell culture versus in your body.
Right.
So, and I'm going to put a pause in that. So let's, I'm going to put a little pin on my paper just to tell me to come back to that. But what did you hear, Angela, of all that?
Oh, my goodness. Well, you lost me at package. Well, I know a little bit about the subject, but if I were just the average layperson, I wouldn't have understood, quite honestly,
Tell me what
much
you
of
understood
that.
from that.
But what I'd like him to explain a little bit more. What is the relevance of exosomes currently in the dermatology space?
We're going to get into that. I'm trying to get into what do people, when they hear stuff like this, understand? What do they take in? Even what you already know. Like, what is it that you're understanding right now?
It's something that is a small particle that is, when released, is assisting in some type of cell-to-cell communication, I think is what I picked up, improving cell-to-cell communication, which could, and that's kind of why I was wanting to get into kind of what is the relevance then to the dermatology space, is like, then what does that mean for the average person who's using products that are advertising exosomes? I
think that actually is a really interesting, that's actually a really good, concise way to describe an exosome.
Yeah, I did a very good job as far as
well, thank
communication.
you.
I was kind of going off in all sorts of different tangents.
Well, the thing is, that's the difference between, technically, what Brian did was something that you have to do to truly understand the nuance. Because what you did was you brought it down to more of a general kind of what does it do. But the fact is, that is exactly the thing that we need to know. It's a cell-to-cell communication, but that actually begs the question of, well, what is it communicating?
Yeah.
Because that is the biggest issue when it comes to exosomes, in my opinion, is that people don't ask that question of, okay, so is it a plant exosome? Is it a mesenchymal stem cell exosome? Is it a adipocyte stem cell exosome? what is this? Are the signals all the same? Absolutely not. I mean, the question is, how do you substantiate that? Because if you ask most people what is the actual signal, they'll have no clue, because that is not what's being sold. What's being sold is the aesthetic benefit, but how is it that all these cells types, from plant to animal and between, because there's a company that does like deer placenta, stuff, and so how is it that people are not asking this question? Because you said cancer cells also, and that's a big
They don't know
thing.
to ask, though, right?
That's the part.
They're buying into the marketing side of the business.
Yeah, but there's a complexity to it. It's not just a single signal that's coming from these exosomes. They're actually quite complicated in the different things that they may have in them. And so again, it's not just this one is saying this particular cytokine and this one's saying this particular growth factor. Again, there may be a complication of multiple different things in there. And again, that signal in itself may be complicated, that missing one piece out versus another may make a totally different signal to the cell type that it's targeted.
Right.
So again, I think that right now it's really in its infancy, and people are taking it for a marketing piece without actually understanding fully what's going on. I mean, there's a lot of research going on it, but unfortunately, marketing doesn't wait for research to fully develop. It goes with where the money is or where the marketing is, and that sometimes is a detriment to actual science.
Yeah.
But great for marketing.
Yeah, I mean, why pay to
Great for sales.
Great for sales.
So why invest in research if you can sell it without doing the research, right?
Right.
And that's one of the things where I would say is actually to humanity in general is not a good thing.
You're right.
Because it stagnates our ability to move forward. And it's just like we were talking about, and as always, I bring it back to the microbiome, it's just like we talk about with that, which is when you make assumptions, then you start building on those assumptions. And you end up in a place that may be completely different than where you should be, because if you're trying to make the skin aseptic, you might think it's okay because everybody's been saying that bacteria are pathogenic or whatever. But when you start to learn that that's not the case, then you have to go back and say, well, where did we go wrong? And then you got to pull the rug out and start over, because you can't, unless you can find which card there. And it's not always easy to just pull one card out, like a Jenga game, And so what I would say is there becomes an ethical question here about how much can you just move forward with marketing with something like this when you're taking it from a source that is not from your own body. Because that's really where, for me, it may not be a safety issue, but you have to ask that question. Otherwise, are you being ethical in the way that you're manufacturing or commercializing?
Yeah.
Well, I think let's be clear, too. In most cases, there is some science, and that's in quotation marks, being presented. May not be good science, but it's being portrayed as science, almost pseudoscience, where the average
or it's just
person...
incomplete. It doesn't have to be
I think
pseudo.
It goes to incomplete, and again, there's a saying that speed kills, and speed kills the competition. So everyone's going out as quickly as they possibly
gets there
can.
first?
Who gets there first? And so I think that's really where people are cutting corners, so to speak. And maybe they're doing the research behind the scenes.
That's true.
But again, we've not really seen a lot out.
And I do want to say, I'm sorry to cut you off, but I do want to say that nobody is going to fault marketing and sales.
Right, we love marketing. And
you don't have companies if you don't have those. The thing is, you have to drive revenue to actually have money to do all the stuff that you want to do. Otherwise, there are grants to go to institutions to do research as well. A lot of companies choose not to do research. They choose just to sell and market. So we can't really fault. That's why those entities exist, is to make money, to give jobs to people and to make money. Now, the thing is, certain companies take it a little too far and do it at the expense of health and safety of people. Other companies do the opposite, where they make very little money, but they spend a lot of time investing in, and that's what usually it's more of a charity or a non-for-profit.
Right.
And it's not regulated at this point in time.
That's true.
So
Let's talk about that.
All right. So again, it's not regulated at this point in time. There's no FDA clearance. It's not considered a biologic, clearance pathway at this point in time. So again, if you look at what the FDA has said, is that they've put out warning letters or cautionary statements regarding the use of exosomes for any purpose.
Not for cosmetic, though, right? Only for indications that are medical in nature, right?
Right, because they're FDA at this point. And that may change. There's some changes within the FDA regarding cosmetics.
That's true.
And so that may, by the end of the year, starting in 2024, that may change, and they may see that as being something that they want to regulate or they want to be involved in.
Right.
So again, at this point in time, you are correct. But there is no, to the best of my knowledge, there is no cleared exosome by the FDA or by any regulatory authority to be used on humans.
But there are plenty of companies selling them.
Oh, absolutely. And there's more and more probably week by week. So.
Right. And the problem is there are some companies that are selling it more in a agnostic way of here are the exosomes, you do what you need to, or making cosmetic claims. And then there's others that are actually quite blatantly, marketing to medical indications.
And again, that goes back to your question about whether it be aesthetics in dermatology. We're looking into orthopedics and looking at benefits there. So again, there's a number of different ways that they're looking at it. But regarding skin, there's quite a few companies that are talking about benefits. And this goes into what would be considered a drug claim and regarding increases in collagen, elastin, and changes in cellular activity, such that if the FDA wanted to take a look at it, they could probably find things that they don't like.
But unfortunately, FDA does not have the ability, or maybe fortunately or unfortunately, they don't have the ability to be everywhere all at once, and so they typically pick and choose the most egregious offenders. And unless there's a huge safety issue, they probably won't drop everything to step in. That being said, let's really quickly, you were going to say you wanted a little more on something from Brian, and so let's go to that.
Well, he just touched on it a little bit, but, obviously we're a multi-division company at Crown Laboratories. So we have our OTC division, over-the-counter division, and our medical aesthetic division and premium skincare. So when you look across the space, dermatology and aesthetics in general, what is the relevance of exosomes? Where are we seeing them currently in that space?
Well, you're seeing them actually in all those spaces currently. Whether you see they're now offered as raw materials and also for formulations to be used, also with products that are actually already out there by a number of companies. Certainly, they're going into aesthetics. Now, it's not so much. I guess I was trying to think in OTC type products. I don't know if there's really.
That would be very. Dangerous at this point.
So topical products, most likely you're seeing exosomes being used in topical products, or?
Most of those are topical products, but they also may be involved in whether they be part of microneedling or some type of source of
there are people injecting them.
Yeah.
Injection
which is where the FDA was
microneedling into the skin, potentially.
Of exosomes into the skin.
Of exosomes in potential, okay.
But again, what the exosomes are, again, that goes back to the question, they're not from the same patient, at least not that I know of anybody that currently marketing a system that goes doing that. So again, they're taking exosomes from some source, whether it be pooled from individuals or they're actually growing up cells in culture, as Thomas mentioned, and then isolating those exosomes and then presenting that into either freeze drying or somehow concentrating those down and then offering those over to either be injected as a material that's neat within the injection or actually used in part of a formulation and then used either topically or injected.
Okay.
Right.
So it reminds me of growth factors, essentially. Is this
well, it's a complicated
20-something years ago, it's the same story, right?
Yeah. We were taking. You got to remember those growth factors were using. They weren't putting growth factors. They weren't like making purified growth factors like cytokines and putting them into a bottle. They were taking conditioned media. So basically, they would grow cells in a media, just take that media and put it into the product. And so it always contained exosomes as well. And so it's very likely that what people were measuring, some of that was the cytokines inside of the exosomes, because we're not going to get into all the way that we do testing, but it could have been that was some of it. Really, it's hard to know. Regardless, I actually would be way more convinced that an exosome would do something than a free-floating cytokine. Because an exosome actually is at least encapsulating it and allowing it to maybe have a better chance of getting where it needs to go. Additionally, there's more than just growth factors that are in the exosomes. Exosomes also can and typically do have genetic material. And that is one of the things that is of importance to note here is that genetic material is not generic. meaning there's different what we call splice variants for genes in humans so we're not like bacteria where every bacteria just does the same exact mRNA. We actually have a series of things that we can cut and paste together in our genes, and splice variant would be different ways that different people can cut genes together. Sometimes it's the same person just doing it different because of a different regulation, like an epigenetic upregulation. And the reason that I bring that in, and I know that's a very technical thing, but the reason I bring that in is because in my mind, when I think about the way cells work, is that if you think about a skin cell, why does it secrete an exosome? Is it to grow hair? Maybe some of it, because you grow hair everywhere. But if I growing, so let's think, for instance, there's a particular company that is very well known that does neonatal fibroblasts from the foreskins of neonatals.
During circumcision.
Yeah, during circumcision. So it's one of those things where, do we think that particular set of skin cells, where a lot of people are saying that exosomes in general can be used for hair regrowth, and so why would a signal from the foreskin of a baby, why would that signal something to increase hair growth? That's the question. Now, of course, it's in a petri dish. It's in a different environment because a skin cell or any cell in its proper environment, in its homeostatic environment, where it's like just basically talking to each other to say, are you okay? I'm okay. Are you okay? Because we do have, we've talked about senescent cells and all that stuff. The fact is the body sends signals for a reason. They're not just things that they just send out willy-nilly all the time. The body does it because they have to communicate or do something for one reason or another. Sometimes cells, as they're migrating, will leave a breadcrumb trail so that other cells know to follow, especially things like metastatic skin cancer or cancers in general. And so there are some times where they send out signals just to say, I'm getting ready to die, so come eat me, or kill me and eat me. And that's what apoptosis is.
Yep.
And so, there's different signals. And the question is, do people understand that in a cell culture, those are not the signals that are necessarily natural to what a cell would put out in the body? Now,
because it's dependent upon the environment,
yes,
right?
And it's an artificial environment that's created to maintain those particular cells. And they may actually have materials in there that may be totally foreign to what's normally seen in the human body.
Yeah, like FBS, like
fetal bovine serum,
which is the plasma or the serum from the blood of calves, which we use in cell biology because it's very nutritious. I guess
it's rich in materials.
We use that to do cell culture, and so the question is, like, for instance, those growth factors have been sold for many years now. We're grown in FBS, so a lot of what people have been putting on them is serum from calves. And the question is, well, is it the growth factors from the calves that were doing it, or is it the cells' growth factors that were doing it? Because there's exosomes in the FBS probably too.
There's pituitary extracts they use in there, which actually do have exosomes in there, so they're actually getting signals from external besides what they're getting. So there's all these complications that come into play as you grow these things artificially.
Well, and when I said growth factors early, what I'm saying is it's reminiscent of conversations you and I have had over the last 10 years of when growth factors in topical products came to light, and there was science presented around those growth factors. As a scientist, I know you looked at that research, and the two things you always came back with is I'm not seeing in the studies, I'm not seeing they don't quantify the number of growth factors, and they don't provide the data that shows me that those proteins are still active when sitting on a shelf for a long period of time.
That's correct.
And I can remember, Thomas, asking you at that point when you shared that particular information with me. I said, well, what would it have taken in those studies to answer those two questions? Because I'm thinking, well, it was very expensive to run those tests, or it would take a long time. And you said, no, it wasn't a factor of cost or time that would prevent a company from answering those two questions. It was simply that was something that was left out of the data.
I think they just don't want to know.
Probably intentionally.
They just don't want to know.
Yeah.
And that's the point that I'm trying to make with the exosomes, is that I think most companies just don't want to, at this point when it's such a hot commodity, they don't want to ask the questions because I think they are scared to know the answers, because then people will say, maybe I should wait to use these.
Or unfortunately, maybe they do know the answers and it's something they're choosing not
Maybe.
to share the
I'm
data,
not sure.
potentially.
I don't think there's anybody nefarious per se. I could be wrong. But I think there are people, if you have a legitimate scientist working for your company, they're already asking these questions.
Right.
And it's not the scientists probably that are not providing the answers. It's probably somebody a little bit higher up the food chain that's basically saying, let's just not disclose that quite yet because it's not required.
Right.
Now, that being said, there's companies looking at, you had referenced using like PRP and the exosomes derived from there and pooling them together. And that's an interesting one because that's one of the few that I've seen that actually is using exosomes that are in the are from a natural environment, if you will, because that's found in a state that is outside of a cell culture.
Right.
So I'm not sure if that's a way to say it that people can get. Did that make sense to you? Okay,
no.
so basically it's free floating in the blood already.
Okay.
And so the question is, if it's already in the blood, that means that it's probably there to either signal something somewhere down the path that something's let it go to let somebody else know.
Within the intact individual.
Yeah. So it's not like in my skin trying to communicate just to the skin. It's actually been somehow put into the blood either on purpose or by mistake, and so that's really meant to find its way elsewhere in the body. So I can understand how somebody might say, for instance, blood is, when you cut yourself, it has a bunch of things like platelets and the different thrombins and fibrinogens and stuff, things that actually are there for the purpose of, if I start leaking, I can set a cascade of motion to do two things: stop the bleeding and then start the healing process.
Right.
And so a lot of those exosomes, in my opinion, might be there for the express purpose of allowing for improved healing once a injury happens. In that case, using exosomes from platelets makes a lot of sense for things like wound healing.
Right.
For like ulcers, diabetic ulcers and such, which I think it has been explored for that purpose. I believe I know PRP has.
PRP has. I'm sure there's somebody that's doing exosome work in there, but in that case, but I don't know if anybody's
isolated them.
Yeah.
So I guess the question is, now we need to know: is it the exosomes that are doing it? Is
Right.
What is doing the actual work there? But it makes a lot of sense to me that might be why this is. What's driving that? But the thing is, what doesn't make sense to me is why people are like, well, they also for hair, because in my thought process is, well, you have blood flow to your hair already, so when those exosomes affect the hair, I mean, what's the difference? And so that's really the question: is there's no substantiation.
Right. Now, again, they may be saying is the fact that they're doing higher concentrations, and so they're giving you a
dose
large,
does matter.
Correct, and so maybe that's why they feel it.
But what's the dose, though?
Well,
that's the thing, is you can say that all day, but you got to give me the dose.
Well, I'm just saying that may be what their mentality is as towards why they say that it works. And there's, they're talking about it towards reduction in blood flow in the scalp areas towards where people start losing their hair.
Right, which is true, right?
So this way it actually kind of overstimulates and gets them to...
But is it the exosomes, or is it just a foreign body reactions causing more blood flow because of an injury?
No.
That's the thing, I'm open to it.
Yeah.
It's all about, it's possible. Totally
Right.
possible. It's just we can't just, in my opinion, it's not ethical to market something that has potential safety issues if you don't know enough to know if it has safety issues.
Right.
And when you're getting things from outside sources, even the pooled PRP, I do have some concerns about, not viruses per se or stuff. I know that they screen for most of that stuff when you give blood in the first place. It's more about, is too much too much? And how much is the cutoff before it becomes deleterious? Because you can't just assume, like if I take an aspirin and it doesn't help my headache, I can take 5 aspirin. But if I take too many aspirins, I run the risk of bleeding out. Having, and it's because you take too much of something, it's going to hurt you.
Right.
And so where is that line, and who is asking that question to make sure that people are not going to harm the people that are being treated with the stuff that has not been tested or studied?
So,
correct. I don't know how to answer.
I am yelling at Brian for no reason.
Agreeing to it. And again, that gets
fault.
into questions, regarding plant-based exosomes, which make, from a toxicologist side, I'm actually more worried about that.
The plants, because of the
proteins that go along with that, and whether or not they really...
Because of the reaction, foreign body reaction?
Correct. And I believe they've actually has some publications on that now, or some granulomas that, based on to your point about injections, and so that's where I guess I'm still worried about the mammalian sourced. But everybody seems to think, natural, and that therefore plants are therefore,
mammalians are natural too, though.
Well, are always all friendly and the like, so because they're natural plants, and that everybody's good with plants.
Well, to argue for the side of plants, and we'll go a couple minutes over just because this is a good conversation, but to argue on the good side of the plant exosomes, because I was thinking about this too. It's like, for instance, the genes that are in these plants, not human genes, but that doesn't mean that they're necessarily deleterious, because there are some extracts from plants and stuff that are beneficial. We know that there's a lot of plants that are beneficial.
Chemicals that are made in plants that also hit receptors within the human body and the like.
So now I don't want to defend them and say that I would say go out and source this and use it willy-nilly, but at the same time, it's all the same in my eyes that there's potential here for sure. There's no doubt about it. I don't think the buzz would be at the level it was if there wasn't enough science to have the potential. What I do think is that it is overhyped as far as what they're saying it can do, and in comparison to what they know it can do, because I don't think they've substantiated most of the claims that they're making.
Right.
And that's a problem for me.
Well, or it's a complicated mix that they're having the exosomes in there with another 10 other ingredients or 10 other materials, versus being isolated and testing only that particular exosome. So again, is it a combination of all of the above, or is it the testing such that they may be knowledgeable or unknowledgeable regarding the bias of a particular study in a way that it's going to benefit their particular product?
It's possible. It's possible. One before, I know you're going to say something, but before you do, just really quickly, at a conference that I was at recently, somebody asked the question about the FBS being in the serum for these exosomes and stuff, and one of these major companies that does this basically said they're getting away from using FBS. They've stopped using it, and I can't remember what they're using instead. But
well, typically they have something else in there, and they'll add, like I mentioned before, pituitary extract, or they get away from fetal bovine serum because, again,
I'm not sure what they chose to use.
Yeah, so
but regardless, they have moved away, so they are changing that. But the question is, what are they adding instead?
And now, how does it modify regarding the exosomes now, as far as the environment that's set up with those particular cells?
How's it changed? Yeah, so is it a different product now?
Yeah, so I guess I would say there would have to be, unless they characterize it first beforehand and now afterwards, they may not know what they have.
And that's to what we talk about with the biome, is that even cells, like bacteria cells as well, the environment that they're in changes what they secrete.
Yeah, that's exactly what I'm thinking about as y'all are talking. Is the same conversation we have.
It actually is.
About microbes,
and interestingly, microbes have been recently discovered to secrete their own form of exosomes. So we won't get into that today. We'll talk about that another day. But what were you going to say?
I was going to say that, this again takes me back to a decade ago, Thomas, when we were talking about growth factors being advertised in topical products. And you talked about them being quantified. You talked about them being active still when sitting on a shelf for a long period of time. And I remember you telling a well-known dermatologist who really liked growth factors in topical products and stood behind them. And you said, you can like the change that you're seeing in the skin from these products, just maybe fall short on linking that change to growth factors in that topical product. Just what I was hearing you saying just a minute ago. It could be any number of factors or ingredients that are giving you that benefit, not necessarily the growth factors, because again, they haven't quantified and they haven't shown that they're active.
Right.
Similar in terms of, because we hear a lot about exosomes in topical products today. So similar, would you say, in how you would approach? Because that's what I was hearing just a minute ago.
Kind of go back to analogies. Whatever, before with growth factors, it was a single sentence. Now with exosomes, you've got an entire book. And so now there's multiple combinations that go into play. And so I think it becomes much more difficult, and that may be why people are not doing it, but actually to characterize them and then to see how the changes in character actually have an influence regarding the tissue. Yeah,
and when he says sentence versus book, he means the amount of information you have to gather to characterize them. Because you're not just looking and seeing what protein is there. You have to see what protein. You have to sequence the RNAs or the DNAs in there. You have to look at the outside of the membrane, see what are what's the proteins on the outside of the coat?
Both the receptors.
The cell receptors. The signal cell receptors, yeah.
But there's still even debate whether or not an exosome contains DNA or not. So again, some people feel that an exosome doesn't include DNA; it's only RNA. Some say that it actually contains small fragments of DNA. So again, there's even debate in the exosome community of what contains the exosome at the very basic level.
Yeah, we're still defining it.
Yeah.
And it's one of those things where it's like we haven't let the scientists kind of define things. We're already marketing it.
Right. So if I'm just sitting back and listening to the two of you, and knowing what I do know about the industry in which we work, is there is some excitement around exosomes. It sounds like there is some science, but there's still a lot of unanswered questions on this topic. That what we're seeing in terms of what's being introduced today into the market in topical products, there's a lot of marketing that's wrapped around that, not necessarily definitive science. Maybe some science, but there's still as many unanswered questions as there are answered, would you say?
I'd say a lot more
unanswered questions.
I'd say it's way more marketing hype than science at this point. The problem is that it's hard to wrap science around this when every company is trying to take a different angle, because some companies may be going about it the right way as far as like choosing the right cell source and all that stuff. Other companies say, I got to be different, so I'm going to go plant, or I'm going to go, deer placenta or something. So the real question is there somebody that is actually doing it the right way, and how are we going to tease that apart? Because one of the things that happens is that if one person does it and substantiates it, everybody says, oh, we do that too, without substantiating it. And just because one group substantiates it doesn't mean that everybody's angle is also accurate. I mean, even in the world of drugs, there's controversy around whether somebody that manufactures a drug and then it becomes generic, whether the generic is actually as powerful because it's a different manufacturing process or whatever. There's a way in which that has to be regulated, but there are people that claim that drugs do have a difference based on where they're manufactured.
Mm-hmm. Sure. I mean, you look at the absorption rate, looking at the
filler or everything that goes into making it could be different, not just the drug.
Correct.
So that's the biggest thing, is that just like skincare, is that you can have a great ingredient and have a lousy formulation. Or you can have a great formulation and an ingredient
never
that's
gets to where it's even supposed to get to. Or is it it's not at the right percentage to
messes
change
up
the skin, or just the opposite, too high of a percentage to actually have an adverse effect
So
on the skin?
One of the things we lose sometimes is we let the marketing drive a little bit too much, I think. Mm-hmm. Again, marketing is essential, because even if you have a wonderful product, you could ruin it. It's not going to go
anywhere.
Yeah.
Or it's not going to go anywhere if it doesn't have the proper
marketing is essential to even getting science out there. But the thing is, when science does not lead, you have to at least ask the questions whether you're willing to take the risks to move forward. And so it's hand in hand. Marketing and science need to go hand in hand in our industry. And I think that's the problem, is that it typically it doesn't.
So let me ask you this. If I'm a provider in the medical aesthetic space, and somebody walks into my office and they present a topical product with exosomes, and I see a beautiful brochure, I see before and after pictures, I see results, and I'm getting really excited about what I'm seeing from this company and this individual, what are the questions I should ask in exploring
as a consumer?
That particular product? As a provider who may actually bring that product into my practice and sell it to my patients. What are the questions I should ask of that company providing that exosome product?
Well, first one is, what data do you have? I mean, a nice brochure is one thing, but what data do they have as towards characterizing the particular exosome? Do they look at stability?
Is this all anecdotal in your brochure?
But again,
so where's the data? So then they present a study or a white paper, potentially. So now I've got the data in front of me. When I'm looking at that data, what should I be looking for?
It's a good question.
Well, I guess I would be looking for a dose response. I mean,
I would too.
I mean, that would be the first thing.
That's not going to happen, though.
Well, that's probably true.
So what I would say is, you, as a physician, you have to make the decision. You are a business person as well as a clinician if you're running a practice, unless you have somebody else doing that for you. And you have to make the decision where your time goes. If you don't have the time, to vet a new technology, I would pass on it until you do. Because really, that's the ethics of medicine there, is that if you are going to charge somebody for for something and you say it's going to do
make a claim.
I make a claim that it's going to benefit. In medicine, there's always a benefit and there's always a risk.
Right.
Nobody ever talks about the risks in aesthetics unless the FDA makes them.
Yeah.
And that's the part that, for me, if you're treating somebody, if I go to a physician, I want them to make sure they tell me the good and the bad, because I want, as a consumer, you are your best health advocate. The physician's not. If they mess you up, unless you sue them, it's not going to hurt them any. It hurts you. So you have to say, as a physician, you have the ethics or the ethical obligation to make sure whatever you do is not going to harm. Do no harm, I believe, is their first mandate. So if you don't have the time or the resources to look into something, I would not use it.
Stand back until you do.
As a consumer, I would not use something unless I felt, and of course I'm a scientist, and so of course I look into things usually pretty deep, but as a consumer, I don't, unless I'm willing to take the risk, I'm not going to buy into something just because of the hype. And typically a lot of times that makes me not an early adopter of a lot of things, because I will wait to see whether people have issues with it.
Yes, you will. Years.
Anyway,
decades.
I think that's a good place to leave this, because there's not a lot of answers yet, but it doesn't mean they're not coming.
Right.
We also are active in looking into exosomes and stuff at Crown, but we're letting science lead that. And so we will be back at some point talking about this, I am sure. But since we got to get out of here and go on to our next one, which is about hyperhidrosis, let's really quick, where are your socials? You got like 3 posts on your Instagram now.
I'm killing it on Instagram. Yes, I'm an Instagram sensation. That's what I tell my kids. I'm at Angela Wilson McDonald.
All right.
And I would love to hear from you.
Okay. And I don't know if you have socials, but if you want to share, or if you want to just...
No, I have no socials, and anybody gets in touch with me, they're wanting me to buy into cryptocurrency. So,
so where can they find you? At Crown's website?
Crown's... Well, I don't know if you know Crown's website.
Main number.
CrownLaboratories.com.
Dr. Brian
I guess
Jones.
I can figure it out.
So if you go to medinfo at CrownLaboratories.com, that will eventually make its way somehow to Dr. Jones. You can find me at dr.t.hitchcock on Instagram, or you can go to crownlaboratories.com. I would like to ask a favor from everybody. We have been traditionally putting the podcast onto Spotify and all those audio websites, and we've got some good traction there. We also been putting it on the Crown website for people to watch the video editions of these. However, we just started a YouTube channel specifically for the podcast. We don't do any marketing, so if you could please share, if you like the podcast, share the YouTube. Go to YouTube, subscribe. Ring the bell so you get notifications whenever we have a new podcast. And if you could like the podcast as well, that'll help the algorithm, especially if you watch one after the other. That'll help the algorithm to actually send it out and recommend it to people. So please, if you could help us with that so we can grow the channel to get more people aware of the Skin Science podcast. And also, lastly, don't forget the book out by Dr. Doris Day and myself called Rebooting the Biome. If you could check that out at Amazon, we would appreciate that. And I think you'll learn a lot from that as well. So from everybody here and those in the peanut gallery that I forgot to introduce, Seti and Alan, thank you for your eyes and ears. We hope you gleaned something from this discussion, and we will see you back here on the next episode. Goodbye for now.